I'm going to also preface quickly, I'm on campus and the internet is surprisingly bad here. So if I'm breaking up or you lose me at any point, I'll be right back. I apologize for that.
I'll just have to entertain everyone by myself, I guess.
That actually happened last week, and my guest was unbelievable. He put on like a stand-up comedy routine.
Okay, I will
just
not
went
do
with it.
that, but maybe I can answer some questions or something.
Yeah. So please, everyone, again, thank you all for being here. And if you have any questions about our conversation, feel free to jump in by throwing it in the chat, or you can turn on your camera and join us on stage or request to join us on stage. But yeah, and then that way, if I get kicked out, then Monique has some questions to answer.
There you go.
So something that we're going to talk about a lot is this phenomenon that you discovered called the social transfer of pain. And it's super interesting. It's one of the coolest things I've ever heard about in neuroscience, I think, personally. Maybe I'm biased. But it sheds a lot of, interesting light on empathy and our ability to study empathy in neuroscience, which is sort of new in like a mechanistic way. So we'll get to all that. Let me just start by asking you about how you got into this field, and if you have any interesting insights or thoughts on the history of this field of social neuroscience and empathy. And again, please, everyone, feel free to jump in with questions.
Yeah, so in all honesty, in grad school, I was in a laboratory that studied social neuroscience, but they were really focused on looking at the social effects on alcohol drinking, using a prairie vole model. So if they were hanging out with their friends, did they drink more alcohol? Did that change which brain regions were activated? But I was not actually looking at any social parameters at that point. I was actually interested in looking at a model of alcohol withdrawal by measuring pain and looking at pain mechanisms in the brain and how those overlap with mechanisms of drug use and alcohol use. But what I found was the social transfer of pain, like you mentioned and you've talked about on your TikTok, where one mouse in pain will just transfer that information to another mouse. So they just hang out for a little bit. And if one mouse has, like, let's say arthritis in their paw or some sort of inflammatory pain state, they socially communicate that information via olfactory cues to the other mouse. And the other mouse, which we call the bystander, ends up developing a pain state that looks indistinguishable from the mouse that we gave the pain to. So anyways, I actually found this phenomenon on accident because I had given mice alcohol and let them drink for a week, which they do readily, which is kind of surprising. And then I took the alcohol away for 24 hours and I saw that they were in pain because hangovers hurt, alcohol withdrawal doesn't feel good, and they were very hypersensitive. But what I found during those studies was the mice drinking water that never had any access to alcohol were also in pain. And there was a moment there as a grad student, I thought, like, oh my gosh, did I give them all alcohol? Why are my control mice in pain? Nothing happened to them. What's going on? And then I basically spent the rest of my PhD trying to figure out why my control mice were developing pain just from being in a social context with another mouse that was experiencing pain. So that's kind of how I got into this field. It was almost an accident, but the more I looked into it and the more we characterize this phenomenon, I realized, no, they're actually socially transferring this pain information. And I started to think when I was looking at neural mechanisms that this is a model of empathy, and the brain regions that are activated are the same regions that are activated in human studies of empathy. And that's kind of what got me on that path. And really, yeah, so it was a totally serendipitous finding. And like you said, I think it's one of the coolest things that I've experienced too. And it was all by accident. I did not start my PhD studies thinking about empathy at all, frankly. So yeah, so that's sort of how it happened. Yeah, are you still there?
Sorry.
Okay.
Yeah, I'm here. My bad. No, that is so cool. I didn't know that story about how you sort of discovered it on accident. And it's also a really great point about how when you have something occur in the lab that seems like a problem, exploring it might lead you to find something really, really interesting. And now it's probably amazing for you to look back and realize that started as something that was probably pissing you off. You're like, why is my experiment not working? And now you have a first author science paper, and I mean a paper in the journal Science, which is arguably, not even arguably, one of the best journals in the world. So it's pretty incredible. Really cool story.
No, and I think it is actually a really cool story, like a cool story about how something happened on accident. And some of the greatest scientific discoveries happen that same way. Like someone had the intent to do something else, but they sort of had the training and the open mind to be aware and sort of find something really serendipitous and really interesting. And to be fair, like it didn't happen quite that easy. It took like 5 years of work to convince my dissertation committee that it was real and prove every other possibility wrong. And then I think it took about, another number of years and my science paper to convince the rest of the neuroscience community that it was real. Which gets to like kind of what you were alluding to at the beginning of, for a long time, the field of neuroscience didn't study the neural mechanisms of empathy in any model but a human model, because there was this idea that empathy is a cognitive state that's unique to humans. But really, I would argue more and more as a field, we're finding out that's not true. And there has been evidence of empathy in animals, like mammals, all the way down to a mouse, that we've seen in scientific literature since the 1950s. But people were just really worried about calling it empathy because they thought that empathy has to be this higher cognitive state that only humans are, uniquely capable of. And it turns out that that's not the case. There are many other sort of forms of empathy that can exist, like all the way down to the rodent. And so even when I was conducting my PhD studies, which was like back in 2011 to 2016, people were uncomfortable with calling this phenomenon empathy. But now I think that I've really shown that in every way we can measure it, it appears to be the same type of empathy that we would see in a human being, sort of without this sort of cognitive, conscious introspection that empathy might cause in a human. But the behaviors and the neural mechanisms seem to match what we would see in a human.
So that's very interesting in itself. I think the study of empathy is so fascinating. But the fact that it's conserved between humans and mice and all other species suggests that it may have some evolutionary benefit. Do you have anything to say about that? Do you have any thoughts on that?
Oh yeah. So I actually have a lot of thoughts on that. I'm glad you kind of brought me back on track here. But no, so I think that, the idea that animals might experience some form of empathy is not actually a really novel idea. It's just sort of novel in the terms of the study of empathy. And now there have been some really foundational studies going back to the 1990s and early 2000s to show that that animals do experience empathy. And when we think about empathy in the brain, what is going on there? So from an evolution, let me back up. From an evolutionary perspective, social species have conserved mechanisms for survival, and socially interacting, having that be a rewarding process, and mechanisms of empathy are actually really beneficial to the species as a whole, because you can learn about the dangers in your environment, or that empathy might trigger caregiving behavior so that others, animals or mammals in your group will care for you if you're hurt, right? Or they might learn about avoiding something dangerous. And so the transfer of that social information and that feeling of empathy is actually really beneficial for all social species. And there's this idea that's been sort of more widely accepted in the last 20 years that the perception of something happening to you activates the same exact neural circuitry as when you observe it happening to someone else. And so this is called the perception action model. And so if the same brain areas or the same brain cells or neurons are activated when you're performing a task as when you're observing a task, we consider that a form of empathy, right? And so it activates the same brain regions, it activates the same neurons, and that's sort of a mechanism that allows for some redundancy in the brain, so that if then you need to learn about that information and use that information for survival, it's a quicker process. You can just observe it and not have to experience it yourself, right? You could observe someone else experiencing something painful, and then you can learn to avoid that much more quickly without actually experiencing it yourself. So that's sort of like the level of empathy that we're talking about in a rodent, for instance.
Yeah, that's really interesting too, because when I think of, for example, if I'm watching a football game, I watch football, and if someone, gets hurt, or, hurts an arm or a leg or something in a way that's gross looking, I hate that. I personally will feel, I won't feel the pain. I won't feel like my leg's broken, but I don't feel good. But on the other hand, and it makes sense, in an evolutionary context, that watching someone else get hurt, it provides a lot of survival benefit for me. But on the other hand, if I watch someone winning an award, right, and assuming I don't know them, I don't feel good. I don't feel as good as they do. And depending on the context, if it was an award that maybe I also applied for, I might feel jealousy. And someone asked, Ping Ping's in the chat said, what about emotion like jealousy? Can it be studied using a rodent model?
I mean, that's fascinating. I'm trying to think of like the, in rodent terms of empathy, we do sort of focus on these sort of negatively transferred behaviors, like, right? Pain is sort of a negative state that gets transferred. And so jealousy is also a negative state, but you were kind of talking about like, you can experience jealousy from watching someone experience something positive. And is that neural circuitry related to the neural circuitry of empathy? And I would postulate yes. I don't know much about like human jealousy research, but we know that like social isolation activates, or like social rejection, emotional rejection activates the same circuitry as if you have like an emotional state from within yourself. Like if you get rejected by a social group, that activates the same regions that if you physically experience pain, for example. So I hypothesize that like jealousy would activate like some convergent neural circuitry that's the same there, but I do not know how you would study jealousy in animals. Maybe make them hungry and then give another mouse food and make them watch eat and then study their brains. I don't think anyone's done that though. It would be kind of cool.
That's actually a really cool idea that hopefully no one in here, no one scoop that, please.
No one steal our idea.
That's a really that's a really interesting idea. And I just had a thought about what you're describing, about watching someone else experience something positive, but now I completely forgot.
Well, yeah, like so if you deprived a mouse of water for a few hours, right? Like it's not going to hurt them, but they're going to want to drink water because they're thirsty. And then they watched another mouse receive water. I bet you it would activate that empathy circuitry that I'm talking about.
Yes, and that you just reminded me. Thank you. So a good example of this, or similar example, is like cringing. When you see someone do something really awkward.
Yeah.
And it's almost like you're embodying the discomfort of doing that yourself, right? It's hard to watch.
It feels bad. It's hard to watch. Like, yeah, I almost like want to close my eyes or be like, I can't look at that. And yeah, that's a feeling of empathy, of like a negative feeling that you're acquiring just from watching someone else experience something bad, right?
Yeah, and now I'm kind of putting this together. Watching someone do something distasteful. For example, if someone goes in the middle of a college seminar and starts dancing on their chair, right? Like that's something that's embarrassing, assuming that they're bad at dancing. And you might feel that cringe feeling, and it without that cringe feeling, without having to internalize that discomfort, you may not learn as strongly from the experience. Because watching that experience and seeing that and thinking, ooh, that's really bad, allows you the opportunity to say, I'm not going to do that in the future because that's a bad thing to do. It's sort of like supporting or reinforcing the learning.
Well, right. And you can generalize that, right, to like other experiences that might be similar. So it really facilitates learning about what you should avoid. And there is evidence in humans and in mice even that if let's say a mouse has experienced the exact feeling. So if you shock a mouse, they've experienced an electric shock. They will learn more quickly and more robustly observing another mouse receive an electric shock than if they've never experienced that at all. So and I think you can think about that like from a human standpoint. I've never broken a bone. So if I see someone break a bone, I'm going to feel like, oh, ow, that's horrible, right? But I'm not going to really understand how they feel. So having had the experience yourself actually strengthens the learning as well. But you can avoid some, like I'm going to avoid jumping off of a desk, not because I've jumped off a desk and broke my leg, but because, you know, I've seen that can happen, right?
Right. Yeah, that is so interesting. So let's move into talking about the specific behavioral phenomenon that you discovered. So this social transfer of pain. You described it a little bit in the beginning, but if you could, you don't have to go into the great details of like how long the mice are interacting and things like that, but just sort of give an overview of what you found.
Yeah, so basically the idea with the social transfer of pain, which is the title of the paper, so you can look those up, is that if you have one mouse experiencing any kind of pain that is induced, and this could be they're experiencing like an inflammatory pain stimulus in their paw, or they're experiencing drug or alcohol withdrawal, anything that they are physically hurting, and that's activation of pain systems directly. If they interact with another mouse that has no reason to be in pain, but they just hang out with them, then that normal mouse, we call it a bystander mouse, will acquire the pain state like really rapidly within, you know, an hour or so. If you just let them hang out together, then you have both mice are in pain. And they transfer not only the sensory sort of experience, so that mouse will look like they're in pain by like avoiding a hot plate or removing their paw from any stimulation. They look like they're in pain from a sensory standpoint, but also from an emotional standpoint. They show changes that indicate that they're feeling really bad, that they're feeling in pain. So they look just like the pain mouse, just from a social interaction. So that's the social transfer of pain that we're talking about.
Now, I made a TikTok on your paper because it was so cool. And one of the comments that I received a couple times, which is actually a really great question, and I'm sure the reviewers ask you when you went to publish this, how do you know that it's not just mimicry? You have a mouse that's displaying pain behavior after interacting with another mouse that's in pain. So how do you know that they're not just imitating it, that it's actually a pain sensation?
Right. Okay. So first, I'd like to say I asked this question really early on. My dissertation committee asked me this, and I thought even if it was mimicry, that would be fascinating and possibly related to empathy, right? Because like, why else would they mimic that behavior? It doesn't really make sense unless there was some advantage. But I did find out it wasn't mimicry, and we did a couple experiments to show that. So first of all, if you block all access to visual cues between the mice, they still display pain behavior. Additionally, we figured out that it was really olfactory cues that they are using to transmit this information. And you can think about olfactory cues in mice as being their main sensory sort of perception. Like we mostly use visual and language, right? Auditory communication. But for mouse, olfaction is super important. So what we did in those experiments is we took the bedding, the soiled bedding that a pain mouse had lived in, and we took only the bedding, so there was no mouse, and we just put that bedding near other mice, normal mice that had never experienced any pain that we knew about. And those mice would develop pain just from being around those olfactory cues in the bedding. So there was nothing to mimic there. There was just an olfactory cue to elicit a response. And you can think about that cue as like any sensory stimulus, like me watching you bang your knee, right? That's a visual input for me to experience empathy. And so this was an olfactory input for them to experience empathy.
Great point. So that's a key way to turn away that comment.
Yeah.
that criticism from a reviewer
would have been cool, but they don't require actually visually seeing anything. And they also show, sorry, I'll just say too, they show a widespread pain state. It's not like they're just mimicking the behavior in the one paw that is hurt, right? So we've done it where we just have an inflammation of one paw. And the bystander mouse, all of their paws hurt. They're sort of experiencing a widespread pain state. And this is something I have never even told you, Ben, but like I'm starting to think like this might be a good model for widespread pain that's not caused by any injury, like fibromyalgia. There's no injury that causes fibromyalgia. there's just a widespread all over body pain. And how does that happen? Right. And so this could also be a model of that.
That is really fascinating. I was just going to ask about, how you think this translates or relates to humans, right? My first thought is, like, if I were to walk around in the emergency room at a hospital and be surrounded by people who are in pain, would I leave there maybe in a bad mood, or, with heightened sensitivity to pain? And then while you're talking, I was thinking about, well, what if I just smelled the emergency room? Because you're talking about olfaction.
Yeah. So, I mean, I think I also have thought about this model as like a model of caregiver burden. We know that caregivers to all sorts of patients, including pain patients, experience really detrimental psychological and physical side effects. So there's been studies to show that spouses of people with chronic pain experience heightened pain sensitivity, more pain that's similar to that of their spouse. So if you have a spouse with neck pain, you're more likely to experience heightened levels of neck pain, right? But and in terms of like caregiver burden, we've always thought about that as, oh, they're just more stressed out. They've lost other opportunities because they're not able to live their normal lives. Like, we've thought of all these like sociological and psychological reasons, but we never have thought about it from, oh, maybe some sensory information is literally being transferred to elicit this change in neural response that's leading to physical pain and depression and anxiety. And so I think we can use the social transfer of pain to look at caregiver burden, to look at pain conditions that don't have anything to do with an injury like fibromyalgia. And so I saw someone asked about mirror neurons in the comments. So maybe we'll just like foray into that now. So that's another thing that I think relates to humans, is that we know that something called mirror neurons exist. And what these are is there are these neurons that fire in the brain when you're doing something, but they also fire, the same exact neuron in the brain will fire when you watch someone doing that thing. And it was back in the 1990s, they discovered these neurons in macaque monkeys. And what they did was they let the monkey grab an apple. Monkeys really like grabbing apples to eat. And they recorded from single neurons in their frontal cortex and in their sort of somatosensory and motor cortices. And what they found was that when they recorded from these neurons while the monkey was reaching, the same neurons fired when they watched another monkey reach, or they watched a human reach for the same apple. And so they show that the same neurons encode sort of self-action versus observing another in action. But so it's been posited that mirror neurons exist in humans, because we do know for a fact that if you put a human in a functional MRI and image their brain, you can show that if they experience pain, that'll activate a certain set of brain regions. Or if they're in an MRI and they watch someone experience the same pain, it'll activate some of the same brain regions. But we don't know if the same neurons are activated in humans. We just know it's the whole brain region. So those studies can be done and still need to be done in humans, but it obviously is very difficult to measure single neurons in the human. And I do think that mirror neurons exist in the social transfer of pain. And I think they drive this and encode the social transfer of pain. But I haven't done those experiments yet. There's something that I plan to do in the future.
That's very exciting. I know, mirror neurons are such a hot topic. People are always very interested in them, and can't blame them. It's such a cool thing. And it makes me wonder, we're going to get on a tangent about mirror neurons. We'll come back in a second, but why wouldn't all neurons be mirror neurons? If any neuron that encodes an action or fires while performing an action, wouldn't they all fire when observing someone perform the same action? Why? It would be very not economical for the brain to have two neurons for every action. So it makes sense that you would encode it in the same neuron. But why would only certain neurons be mirror neurons and others not?
Good question. So you're right. I think people that argue that mirror neurons don't exist, why would your brain have specific neurons for observing versus performing the same action? You want to condense how much activity your brain needs to function, right? But I think that they have to be neurons that are receiving all this convergent information, right? I'm taking in a lot of information. This is, I have to activate memory, right? To remember, maybe like when I've done that thing, I have to activate somatosensory input of like, and sort of imagining my own self doing something, right? Visual input, perhaps visual and auditory and olfactory. So there are all these neurons that are encoding different parts of the process. And then there's some point of convergence. And I believe that's the mirror neuron, is where all this somatosensory and memory input are converging onto a single set of neurons, or maybe neurons within a few brain regions. Does that make sense?
Interesting. Yeah, definitely. And there's a very good comment from a incredibly intelligent man in the audience named Doug Rein.
Oh, is this your dad?
It's my dad. He said, if all neurons were mirror neurons, then watching someone break an arm would cause you to feel the same pain. And it's a great point, because that would be very bad. You know, if you actually had to feel the full-on pain of someone breaking their, or you breaking your arm when you watch someone do it, that would be terrible. But feeling a bit of it would be helpful. And that is where social transfer of pain sort of fits into all this. So why don't we talk about the neural circuitry, the neurobiology of social transfer of pain? Because after all, this event, I've named it the neuroscience of pain and empathy. So let's talk about what you found neuroscientifically when you started looking into, you know, what was going on in the brains of these mice while this was happening.
Sure, yeah. And I mean, so like I said, there's some level of convergence, right? And when you experience pain, so imagine I experience a painful stimulus on my hand, that activates sensory receptors that project to your spinal cord, and then that information has to travel up your spinal cord into your brain. And then there's a bunch of processing that happens there, and your body can either decide to inhibit that signal and maybe provide endogenous pain relief, which is something that can happen. Or if you're in a state of stress, it might exacerbate that signal. There are different ways in which we can sort of cognitively modulate our pain state. So that's the basic pain processing pathway, right? And so we know when we induce pain in an animal, say we put some inflammatory stimulus into their paw, this is the pathway that gets activated. It's called the ascending pain pathway. And then the one that controls pain is the descending pain pathway that goes from your brain back down your spinal cord. And so with the bystander mouse, the one that socially acquired the pain, they don't have any activation of the sensory receptors, right? Nothing activated pain receptors in their periphery. So how is that information getting in? And so I believe that the point of convergence that I was sort of talking about, like, so when that pain signal in a pain mouse gets up to the brain, it's got to go through the brainstem, through this area called the thalamus, which is like a sensory relay station, up to the cortex. But I believe the animal that's watching the animal in pain, that ends up acquiring the pain, they have this olfactory input that goes from the olfactory bulb directly into the cortex in those same neurons that would be activated in the pain state. And then the descending pain pathway is activated. And so there's this region called the anterior cingulate cortex. And we know in humans, the anterior cingulate cortex is activated during pain and also while watching someone in pain. In fact, it's the most commonly activated region in studies of empathy in humans, in like a human fMRI experiment. And we found in the social transfer of pain, in fact, the anterior cingulate is highly activated in the bystander mice. And if you sort of inhibit activity in that brain region, shut it down, they cannot acquire the social transfer of pain. So that shows us a couple of things. It shows us that this really does look like empathy regions or empathy in the human in terms of neural activation patterns, and that there might be a place where we could prevent that social transfer and that sort of acquiring the empathy, right? So yeah, so here's the ACC. You can see it's like this big swath of cortex, and it's known to be important to emotion and pain as well as empathy. And so it's really essential for the social transfer of pain is empathy.
Yeah, so I just wanted to pull this up to help the viewers understand what part of the brain we're talking about. So I'm not sure, I don't believe that you can see my cursor, but you can see there's this kind of right in the middle of the brain, there's this white sort of upside down U, and then right in front of that on the right is the ACC. So it's this sort of deep area in the cortex. And it's so interesting that what you're saying about how this part of the brain was activated in the mice during the social transfer. And it's also the most activated part of the brain in humans experiencing empathy. I feel like that's very telling in itself, because some people might argue and say, well, it's a mouse. Can they really experience empathy? And that piece of information alone suggests that if it's not empathy, it's something very, very similar, and it's at least encoded by the same part of the brain.
Right, exactly. And what some people might not know is that mice share about 90 to 95% similarity of their brains compared to ours. So they have almost every single brain structure that we have, and all of the structures that encode pain and social behavior are the same between mice and humans. And so they may not have this sort of cognitive perceptual experience that we have during empathy, but they do experience a basic form of empathy that, requires the same brain regions, including the anterior cingulate cortex.
So I have read your paper many times, and of course your Science 2021 paper, and I know that there is another brain area, at least one, involved in this as well, the nucleus accumbens. So if you could you describe a bit about, first, what this exactly means, like describe the circuits involved, and then also if you could talk a little bit about like how you got to this discovery in the brain. I feel that it's such a huge jump to have, you're watching a mouse do something, and then suddenly you've pinpointed the exact brain area and actually the exact neurons in the mouse brain, which are active during this experience. So if you could sort of walk us through that process of discovery. I'm going to move us back to the meet and greet stage.
Yeah, sure. So yeah, you're right. I saw this behavior and I sort of made sure this was a real behavior and I didn't mess up my experiments, and this was something I could replicate, because it's super important for scientific experiments is to replicate it and make sure you're really getting at the right question, right? So once I decided, like, yes, these mice, they socially transfer their pain, and then I really wanted to know what is happening in the brain. Does it look like human empathy? So how did I do that? And so mice are actually pretty cool to study because we have lots of control over their environments, and we also can measure their brain activity and manipulate their brain activity in a bunch of different ways. So one way you can do this is after the experiments are over and you want to analyze their brains, you can stain the brain for something called CFOS. CFOS is basically just a marker of neural activation. So I can just ask, what neurons were activated during this behavior. And I can look for this CFOS staining on a brain slice and count the number of cells that were activated. So that's one of the first things I did is I counted all the cells that were activated during the social transfer of pain in the pain mouse and the bystander mouse. And I showed the anterior cingulate, the nucleus accumbens, another region called the insula. All of these regions were hyperactivated in the bystander mouse. So that was kind of like my first step. And I was like, okay, well, this is really cool. This shows the same regions that are activated in human fMRI studies, right, are the same regions that are activated in the mouse. But then I wanted to really show a causal link and sort of look at manipulating that brain activity. So there's something that we can do in mice called optogenetics. And basically, we can insert an ion channel into neurons that is activated by light. And what I mean by this is if you've ever seen, oh my gosh, I'm going to blank out on what it's called. The fluorescent algae in the ocean, that when it moves, it fluoresces, right? And so these algae have been reverse engineered. Instead of movement causing them to fluoresce, people have brought these into the lab and genetically modified these ion channels to say, you know what, I want this ion channel, which makes a cell activated, to open in response to blue light. And so you can put these ion channels into the cells of a mouse by injecting it into their brain. and then you shine light on it. So you really put this little LED light into their brain and shine light to activate the neurons. And so I could activate the neurons in the anterior cingulate cortex while the mice were acting, were behaving. And so you can either activate or inhibit neurons in real time using optogenetics. So it's a really cool technology, and you can control very specific populations in the mouse when they're just actively behaving, while they're eating, while they're drinking. And so I decided to inhibit by shining light on the anterior cingulate cortex during the social transfer of pain. And what I saw was if you inhibit the anterior cingulate cortex during this social interaction, the mice no longer acquire the social transfer of pain. So that tells me that the anterior cingulate cortex is required for that behavior. And as you mentioned in my science paper, I showed the nucleus accumbens is also activated. And I showed that by looking at CFOS activation patterns, and that I found the anterior cingulate cortex projects or connects to the nucleus accumbens. And this is really cool because the nucleus accumbens we know is essential for rewarding behavior. Lots of dopamine is released there and is activated by lots of different behaviors, including aversive or negative behaviors, as well as like extremely rewarding behaviors. And we know that the nucleus accumbens is essential for social reward from some other optogenetic studies that have been done. So what I did now is I looked at the neurons that project from the anterior cingulate cortex to the nucleus accumbens and specifically inhibited those. And there are some genetic tools that we can use to do that. And so I shined the light in the nucleus accumbens and showed it's really these anterior cingulate neurons that project the nucleus accumbens that are responsible. Because if you inhibit that pathway, these mice will not acquire the social transfer of pain.
I have to commend you because for a real life scientist who runs their own research lab, you do a phenomenal job of explaining these really complicated topics to a non-scientist
I hope
audience.
others agree. Hopefully that made sense. Yeah.
Yeah, and it's so much that I feel like I owe tuition.
Well, you can Venmo me. No, I'm just kidding.
So I just have a few things to comment on all of that. It's so interesting. I mean, so first, to summarize, you've said that the anterior cingulate cortex is part of the brain which is important for empathy in humans. If you shut off those brain cells while a mouse is interacting with another mouse that's in pain, you can prevent that bystander mouse from taking on that pain behavior, from appearing to show pain afterwards. So in a sense, you're blocking empathy, maybe, if we're considering this empathy. So there's a paper that I made a TikTok about one time where they gave people ibuprofen and then asked them to watch people go through painful experiences, both social, social embarrassment kind of pain, and physical pain, like clips of people getting, like punched or something. And the people who took ibuprofen reported not only that the experience for the other person was less painful, but they also reported that it was less uncomfortable for them to watch it themselves. And it all ties in together, that this idea of if you can block the parts of the brain that you would be experiencing the pain through, then you can also block the pain in yourself. And I just want to ask you, while we're on this topic, do you think that actually, this type of thing could be a real therapy for helping people in some way, like blocking empathy to reduce pain?
Yeah, no, so I think it's really cool, right? Because you are seeing that those neurons that, encode pain in yourself, if you like preemptively block those before the experience, they dampen your ability to experience empathy, right? Or your ability to like truly determine how that other person is feeling. And so you're kind of getting at something that I think is really interesting that someone else asked in the chat was, do you think they would experience similar social transfer for emotional pain as they do for physical pain? And so I did show that they show negative emotional states in this process, and that I'm actually currently looking into whether or not it's the same or different neural circuitry that is involved in sort of the emotional aspect versus the physical aspect of the pain. And I think that if you could block these brain regions like in humans, or use this sort of like in a psychotherapeutic way, I think it could be really important for people who have trauma, who have emotional trauma, and they might have emotional trauma just from things that they've seen or experienced, right? And so if you could find a way to sort of, and like if you think about trauma as activating these neural circuits or overactivation of neural circuits that's maladaptive, and you could dampen that, I think you could really help people with emotional trauma. Or, and there's other types of like physical trauma you can think of like that. For instance, like phantom limb syndrome, where you feel pain, you feel literal pain, and you don't even have the arm there anymore. So for people that don't know, if you have an arm or a leg amputated, it's actually pretty common to feel severe pain where that arm or leg would be, even though it's not there. And so this is sort of like this maladaptive circuit mechanism in the brain. And if you could dampen that, I think it would be really powerful.
So there are several people in the queue. And I mentioned this at the beginning. So there are 7 people in the queue right now. And if you are one of those people and you don't know you're in the queue, that's perfectly fine. What that is essentially you're waiting in line sort of thing to join us on stage and ask us a question or say something, make a comment. So I just want to break for a second and ask if anybody that's in the queue or that is not in the queue would like to turn on their camera or just their microphone and ask any questions. If so, I believe that I will get like some sort of notification if you do.
Okay, well, fun shout out. My grandma, Janet Lytle's in the queue. Grandma, did you want to ask a question? I think she might be there on accident. I'm not sure.
Well, huge shout out to your grandma for being your grandma and figuring out how to get onto this app.
Right? I'm so proud of her. I didn't even tell her how to do
it. That's extremely impressive. Your grandma is very technology literate.
Technically savvy, yes. She does Zoom
Yes.
calls with her friends.
Oh, that's so cool. Okay, so assuming nobody in here, like if that changes, please feel free to turn on your camera. Again, also you can also chat. You can send questions in the chat. So I want to point something out. When you were talking about optogenetics, this is sort of an aside. People ask a lot in the comments on my videos, why do we use mice? And there's questions about cruelty and pain and all this. And we can discuss that if you'd like. I just want to point out, using the tools that you were describing, we can do such amazing things. Like we can actually expose a mouse to a certain experience, good or bad, or neutral, and capture the specific brain cells which were activated by that experience. And then if we want to, we can specifically manipulate those exact brain cells by either turning them on or turning them off to see whether that changes the behavior. Or we can say, we want to know whether those brain cells in that certain brain area, if those connections with another group of brain cells in a different brain area are important versus multiple other brain areas. We can legitimately track down specific neuron-specific circuits in the brain and how they relate to behavior. And that is such an incredibly powerful tool for helping us understand neuroscience. In this case, empathy, pain, but also in other things like neuropsychiatric disease, Alzheimer's, things like this, which really allow us to advance neuroscience as a field much more rapidly than we can with non-invasive sort of observational studies in humans. So I just wanted to point that out.
I couldn't have said it better myself, like about we can use all these different sort of genetic, because we have the mouse genome completely mapped, and we can target cells in a genetic specific way, in an activity specific way, like you mentioned. If they're activated during one experience, we can sort of capture those cells and then mess with them later, turn them on, turn them off. You can see what types of genes those specific cells express. And these are all things we could never do in the human, because it's like, right, it's highly invasive, and you need the tissue at the end of the study to look at those neurons. And so we do have some really powerful ways to look at brain activity in the human, like so an fMRI or a PET study. So that's positron emission tomography, like things like this, but they're all correlative. And we can't get any causation from that, right? And so, yeah, having the mouse model is really key to understanding how all these sort of different behaviors and different genetics really play into this, and what specific neurons are involved.
Yeah, exactly. And I just think it's so powerful. And a lot of people don't realize that there's such a massive gap in the type of questions, scientific questions that we can ask in humans versus in mice, and, you know, even fruit flies and roundworms.
Well, for instance, too, like, and because the other question is, why can't we do this in a dish? Why can't we do this with a cell line or with worms? And that's what I'm saying is, you need a social species to see these actual physiological changes in the brain that are important for social species. And so that's not going to be true in a cell dish or with a line of cells or something like that.
Yeah. So I want to get to a couple of the questions in the chat. Somebody asked, I wonder if doctors and nurses are impacted by this, spending so much time viewing pain and disease. And another person asked, what do you believe the implications could be for therapists who sit with clients in pain for hours every day?
Yeah.
I would love to hear your thoughts on those.
Yeah, no, I mean, so I kind of mentioned this at the beginning, that I do think this could be a model of caregiver and even medical worker burden and burnout, right? We're seeing a lot of that now, that the burnout is so high, and it might not just be stress or anxiety or lost opportunity in these sort of psychological, sociological ways we've thought about the impacts on healthcare workers and therapists. There might actually be a physiological mechanism in which this social communication is leading to changes in the brain. And so, do I think that there are ways that we can combat that and protect these people? Absolutely. But what we need to know is what is actually happening at the level of the brain. We need to know that first. So then we can kind of work backwards and say, what do we do about it? How do we prevent it? How can we target those neurons? How can we protect those neurons, right? How can we protect the brains of those people so that they don't develop all this burden, anxiety and pain and things like that? And so, yeah, I think that there's a real possibility this directly translates, especially to medical workers.
Yeah, I think that would be extremely helpful if those types of sort of quiet issues, you know, you don't really think about that a whole lot, and it's not typically addressed as a critical issue, but it is a burden.
Yeah, and I think it's going to become more of an issue now too, right? Like with COVID and everything, the medical worker burden is going to become a huge problem because, you know, we're losing a lot of healthcare workers. So it's something we need to consider. Can we model that? Can we find treatments to prevent that from happening? Right?
Yeah. So is Janet your grandmother?
Yes, that's my grandma.
Janet,
she wants to know about PTSD.
Yeah, if you'd like to join us, please. I'm gonna feature
turn
you.
Your camera at the top.
I'm gonna see what happens when I feature her. Welcome to the stage, Grandma Smith.
Grandma, we can hear you.
Why is it freezing?
Well, we can still hear you. Nope. Now we can't hear you.
Okay, I think she's. I'll bring her back.
She can help you later.
Janet, you have another opportunity whenever you want it to join us on stage. I'd love to hear your question. Yeah. So, what was I just going to ask you about? Oh man, I really struggle with memory loss, don't I? So,
did you have COVID?
I don't think I did. Oh, okay, Janet is back, but I do have brain fog. I think it's just natural.
Oh, okay.
Let's see.
Just being a scientist. It's life.
Yes. Okay, I'm unable to feature her right now.
Okay.
There's a green check mark. Can you do it? Are you able to?
No. No. I can remove you. I can remove you, Grandma. That's it. Okay.
Strange. Okay, I remember my question. So Alyssa Lopez asked, is this
Hi, Alyssa, that's one of my students, by the way.
Oh, well, fantastic question.
Yeah.
Is this social transfer with mice the same as humans who believe they are empaths? And I want to add, I was going to ask you about this earlier. My mother, she's probably here, is a big empath. She considers herself an empath, and you know, I know she is. I know that scientists like Eric Nestler, for example, have looked at if you stress mice or rats, you can divide them into sort of stress susceptible or stress vulnerable and stress resilient groups. And that just sort of occurs naturally. So have you ever thought about dividing your population of mice into those who are strong social transfers and those who are not? And then I guess if you'd like, please tack on any additional info related to this question.
Okay, so I'm actually getting a lot of good experimental ideas. There was one from the beginning. I can't remember what it was. We're going to have to rewatch it. But this is a fantastic idea, because what I do see is that the bystander mice, the ones that acquire the pain, they actually, some of them are less sensitive and the others aren't. And I've never split those into two groups to see what's different about them, but there has to be something different about them, right? And so if we could figure out what makes them more susceptible to this, we might understand people that are more susceptible to empathy. And I think there's a lot of evidence now to show that there can be some sort of adaptation in certain people, and we don't know if it's genetic or what really causes it, but that makes them like empathic to a maladaptive degree, right? Like I've heard some stories, I think on NPR, about like a woman who's afraid to leave her house because if she sees anyone going through anything emotional or painful, like she cannot deal with it, right? And so like when the level of empathy becomes detrimental to your quality of life, like imagine that we could figure out where that is in the brain and then fix it. Right. And that's not something that I think a lot of people have thought about. They've thought about that as primarily like psychological, sociological. I'll go off on a tiny bit of a tangent here too, is because females, there's been a lot of studies to show female identifying people might experience higher levels of empathy. And whether or not that's the way we define empathy or how we allow females in our society to display empathy is one thing, but what if there really is sort of physiological mechanism for that, a neurological mechanism that we can't just write off by saying like females are hysterical, right? Like let's find the mechanism and figure out if there really is a sex dependent difference there, and like what do we do about that, right? For people where it's actually affecting their life.
Yeah, that's so interesting. I want to come back to this conversation in a moment, but Janet is here and the video is working.
There
Welcome
she is.
to the stage.
Hi,
Can
girl.
you
How
hear
are
me?
you?
We can hear
Yes.
you and we can see you.
Hi.
You're great.
Well, that's scary. Well, this is very interesting. I traveled all the way through Monique's studies. I would go see her in Portland once a year while she was going through this, and I actually understood what she was doing. So it's been very interesting to me throughout this whole process, and I'm really enjoying this today. I'm really proud of her. It makes me cry. And I'm full of empathy. So that did trigger a question. I mean, would there be an application of your studies that would have to do with these sufferers of PTSD? There's so many of them. Something that could shut off a certain part of their brain to help them not suffer so much?
Yeah. So no, that's a fantastic question, Grandma, because one of the models of empathy in the rodent is called observational fear. And that's actually where one mouse watches another mouse get an electric shock, and then they acquire the fear behavior, which mice freeze when they're afraid. And so they will just start freezing if they see something happen to another mouse. And that's actually our primary model for PTSD in the rodent, to understand how PTSD works in the brain. And actually they've developed new treatments for PTSD, including MDMA, so ecstasy assisted therapy. They've shown that MDMA administration in observational fear in the mouse can prevent sort of the long-term memory of this observational fear behavior. And so developing the social transfer of pain model into a model of PTSD could be really, really fascinating and interesting, because, you know, people don't only develop PTSD necessarily. It's not necessarily tied to fear. They can develop post-traumatic stress disorder from a variety of different experiences, right?
Right.
So how do those brain mechanisms sort of differ, I think, is a fascinating question. And so people are using a similar paradigm to study empathy and PTSD, but I haven't done it yet. So, I mean, I probably should. That's a good way to write my next grant.
My grandma gave me the idea.
Yeah, I'll put you in my acknowledgements.
Put it in the
Yeah,
acknowledgements.
acknowledge.
Yeah. Well,
Right.
thank you so much for joining us.
My pleasure.
See you later, alligator.
Thank you.
So that was fantastic. Thank you
Thanks
for joining
Grandma.
us, Janet. So I want to go back to, because I had this amazing idea that I'm actually really, really kind of excited about, and I want to know what you think. We're talking about sort of classifying the mice by whether they are very empathic or not empathic, and like you were just explaining, trying to get to a sort of a neurobiological understanding of what's going on there. But I started thinking about the other way around and looking at those who experience like no transfer, and I wonder if that could be a model of like studying sociopathy. Exactly.
Yeah,
it could be really interesting to poke around in that, because as far as I'm not familiar at all with the literature on this, but I would assume that there's very little. Do you know anything more than I do?
I mean, so one of the labs in the Netherlands, the Kaiser's lab, that is, they do the observational fear paradigm to look at empathy in rats. They've done some human imaging studies. There was this grad student, super brave grad student, by the way, who went into prisons and put psychopaths, people with like social personality disorders, into an fMRI and looked at which brain regions were activated during empathic experiences. So when they watch someone in pain or something like that. And what they found was, if they didn't preface the experience with anything, they just had them watch, the same brain regions did not light up as when they experienced pain themselves. But if you prefaced it by saying, we expect you to experience empathy during this experience as if you experienced it yourself, then the same brain regions would light up. So anyways, there's this really interesting cognitive control that psychopaths have over the ability to initiate these circuits. But I do think in general, we can learn about empathy deficits by looking at those mice that don't acquire it as strongly, right? And there's not just empathy deficits in psychopaths. There's empathy deficits in a lot of neurodevelopmental disorders, or during cognitive decline and dementia. There can be a lack of social understanding, or during autism spectrum disorder, there can be sort of and I'm not saying empathy in a sympathetic way. People are still capable, that have autism spectrum disorder, are still capable of being sympathetic, but sometimes they don't understand social situations the same way, or they don't understand people's facial expressions as exhibiting something, right? And so if we could look at empathy deficits in an animal model, then we might be able to sort of understand that more in terms of neurodevelopmental disorders or personality disorders.
Yeah, it's interesting. I was thinking when you were describing earlier, which by the way, I thought was really cool, about how when you experience pain, it's ascending up from the sensory input to the brain, where in this case, it's sort of descending, coming from the nostrils, olfaction into the brain of a mouse in this case, and then sort of back down to affect the sensation. But I was thinking about this question of the scent comes in through the nostrils, enters the brain. How does it, obviously the scent itself isn't like going and attaching to brain cells, but how does the brain know like what parts of the brain to choose to activate? You know what I mean? And we could talk about that if you'd like, but I'm mentioning it here because in this sense, in something like autism spectrum disorder, there may be some sort of disconnect in that process of registration of social information and accessing and activating the brain areas. I don't want to say appropriate brain areas, but the brain areas that would be activated in someone who does not have autism spectrum disorder. And I think I'm always fascinated by that question of like, how does information just sort of enter the brain and like find the right neurons to activate? It's such a wild question.
No, but I mean, I think like part of it, which is definitely not the whole explanation, is what we were talking about earlier, which is if you've experienced something, you've sort of already created that neural pathway for that. You've learned that, right? So when you observe it and it activates that same pathway, that's sort of an easy connection for your brain to make because it's already learned it. It's stored in your memory. It's something that you've created those synaptic connections for, right? And so whether that's me observing you through visual input is similar to a mouse observing it through olfactory input, right? The same way that a smell can initiate a memory, it can also like sort of access that pathway that's already been accessed. And so my idea is that, whoa, I just lost my train of thought, is that if you've experienced it yourself before, it's going to be a lot easier to activate that pathway. Does that make sense?
Yes, absolutely. And I want to take a second to pull this picture up. Let's see. Okay, I hope people can see this in a moment. So I just wanted to show, so this is a picture of a mouse brain. Frankly, I don't know what's going on in the bottom, so pay no attention to D, E, and F. I just grabbed this from the internet. But if you can see on the top, we have 3 images of the mouse brain. And you can see the large area that takes up most of the left side of the image is the brain itself. But then those 2 balls at the front of the brain, those are the olfactory bulbs. So this is just an image to show you how much of the mouse brain is devoted to just sensation, to just sensing odors that come in through the nose. It's actually quite amazing. And in the human, the olfactory bulb is like tiny in comparison relative to the massive size of the brain.
Yeah, it is. But our visual cortex is gigantic, right? The part of our brain that interprets visual information is more represented because that's our main sense, right? But yeah, where for a mouse, the olfactory cortex is so much bigger because it's really important. And it would be the same for like a dog, for instance.
Mm-hmm. Yeah, and I want to take this opportunity to answer this question, the most recent question. Any thoughts on social neuroscience in the workplace? I feel like with remote work, there's less opportunities for empathy. And I just want to point out, before I allow you to answer this, it's funny because if mice had to go on Zoom, they would probably suffer a lot more because olfaction is such a key component of their social interactions, where humans are much more visually engaged. And so being in a Zoom call may not be so much worse for humans because when we're together, I mean, how much of my social interaction is relying upon smelling the person I'm interacting with? You know, very little, I would hope, and I'm definitely not doing it intentionally. It would be sort of a subconscious process. But yeah, like you said, that the visual cortex is so enlarged, it plays such a huge role. So what are your thoughts on this, the social neuroscience in the workplace idea?
Yeah, so I think that that auditory, so language cues and visual cues are much more important for humans. But that sort of comes so I think that Zoom does, or like being on the internet on a video does change social interactions, but I'm not sure it's in the same way. It's through empathy. I think we're probably still able to experience empathy on video, but there's just much less social interaction overall. So you're not having all of those social interactions that are rewarding, right? You're just sitting in meetings for the most part. And so it's a lot easier to be rude to someone in a meeting if you don't run into them in the hall and have those rewarding social interactions. And so I think that is the problem. But this is something I speak about with my students a lot, that, you know, when we're wearing a mask, that actually takes away half of the social information of the face that we're receiving. And there has been some studies to show that actually the sort of micro expressions we get from the eyes are more important than the mouth. But our brain actually looks at the face as a whole unit, not individual parts. So wearing masks, I don't think it's preventing empathy by any means, but I do think it is making social memory more difficult. I don't know if you've had this experience of like not remembering people's names as easily while they're wearing a mask, and it's because you don't have their whole face. But anyways, that's sort of a tangent that's not related to empathy. But I think the pandemic is affecting a lot of things.
Yeah, there's a person who joined the lab and I met them. I knew them for probably 3 or 4 months, only in a lab setting with a mask on. And then at some point I met them outside the lab and they took their mask off and I did not recognize them. I was like, that's what the bottom half of your face looks like? I can't believe it. It's unbelievable. I pictured it totally different. And I know someone who trained themselves throughout the pandemic to, when wearing a mask, to just sort of squint. I can't do it. Obviously, I look like I'm in pain when I'm doing it, but they squint their eyes to look like they're smiling. And so they're just.
Oh, I do that too. I try and do that with my students. I'm like really like do something with my eyes. So I'm like, yeah, I swear I'm smiling under here, right? Because you're trying to communicate that you're smiling with your eyes. But the good news is we actually get a lot from micro expressions that we're not even conscious of. And a lot of that is around your eyes and your forehead. So I think we're good.
Interesting. I'm just looking at this. This is a great question from Aaron Jones. Have you ever considered doing the social transfer over a longer time course, like pairing a bystander with a mouse in pain for several months? Is this one of your students?
This is
a really
one
good
of my
question.
Students. Hi,
I
Aaron.
knew it.
Yeah, you gave yourself away with the detail. It's a fantastic question. Actually, I don't think I've ever talked to Aaron about this though. So it's a good question is if you leave, so I've been talking about social transfer of leaving the mice together for an hour and they acquire this pain state. But what if you left them together for a really long time? Does that bystander mouse like maintain pain forever? Or is there some habituation, right? Because if the idea is they're acquiring this pain state to learn something about their environment, wouldn't they eventually learn there is no danger and they don't need to activate this neural pathway? Well, that would be my hypothesis, is that there is some habituation to this. There is some control that they can say, look, this cue, this social cue is not predictive of anything that's actually happening to me. So I'm going to shut that down. I'm going to ignore that. Right. And so that would be maybe more representative of how healthcare workers are still able to function when seeing some pretty gnarly stuff. Right. They have the cognitive control to like not emotionally react. And so, that's that question, that last one. Did you see that? So I would hypothesize that, yeah, actually there is some habituation to inhibit this mechanism based on cognitive control and learning. And so, Aaron, it's a good experiment. Maybe we can do that.
It is a great experiment and a great answer. It makes perfect sense. I do kind of want to get into this Neuralink thing, but I want to touch on this idea you were talking about of you don't think Zoom affects empathy. It's funny because I was talking earlier about this idea of watching a football player get hurt and I feel the pain, right? That's happening through a screen, right? Or if I watch a video on my phone of someone, enduring pain, I feel it for sure. And that doesn't really go away either in a Zoom setting. I think it is sort of funny. The anonymity of social media kind of makes you more, not me, I'm not this way, but it makes a lot of people more prone to being mean, essentially, to not being empathic.
Totally inappropriate.
Yeah, and to discarding another person's feelings and just sort of, attacking them. And it's funny, I just want to bring up this example because it just happened the other day. Someone made a video basically attacking me on TikTok. They posted my video and then they were like, this guy cherry picks data and he's a narcissist. Everyone in neuroscience is a narcissist. And, it was just a really mean video for no reason, basically. And I commented on it and I was like, wow, you're really coming from my head. And I kind of addressed a few of the points they were saying that I give really surface level information. And so I was basically saying, oh, well, it's TikTok. I'm sorry. I agree. I wish I could include more nuance.
I've only got
it's very
60
important.
seconds or whatever. What do you want me to do?
Right. And so I just kind of commented friendly. And she replied and was like, I'm so sorry. I didn't think you would see this. I didn't mean anything I said. I was just in a bad mood. And it's funny how even when I become real enough, where it's just a comment, it's just a line of text that I stand behind, suddenly they feel empathy. Whereas when I'm just a video, I'm just a distant person behind a phone screen that doesn't necessarily truly exist in any interactive form, I'm much more vulnerable to just, a baseless attack that they don't even necessarily stand behind. So it's just kind of funny how the closer you get to a true face-to-face interaction, the more prone you are to feeling empathy, and the further apart you are, the more vulnerable you are to attacking someone else. I think it's an important point for everyone to know about the internet.
And I think it takes multiple cues, right? And when you're just looking at comments on Twitter or whatever, you're not associating that with a person with feelings, right? Because you have to think pretty hard to connect all of that because your brain's not used to that being sort of the cues you're used to when interacting with a person. But I mean, there are all sorts of issues with social media in that realm, right? Of just like this overwhelming amount of information that we need to become desensitized to, because it's not the amount of information, emotional information, social information that we are sort of used to as a species receiving on a daily basis. And I don't think we're totally capable of dealing with that much information.
Yeah.
But yeah, I'll say like, even like my husband played a video earlier today, which on whatever, that was like a baby crying because of some stuff going on in the world, and that I didn't see anything. It was enough for me to be like, oh my God, please turn that off. That is really like triggering. I cannot listen to that in the context of what you're reading about right now. Do you know what I mean? So I think we still are capable of feeling empathy with limited information, but we have to give it like a little bit more of our attention and forethought.
And that's also interesting because it goes back to what you're describing about if you experience the pain first, it makes it more severe. So I will tell everyone, I hope this is okay for me to share. So you are a mother. And
yeah. Oh, and I never had that problem before I was a mother. Like, whatever, I could watch movies with babies that were unhappy. Now, like, not a chance. Like, not a chance. I had a full-on, like, anxiety attack this morning. I was like, turn that off. I'm in such a bad mood now. Do you know what I mean?
Yeah.
And so, yeah, like, if you have sort of those neural pathways activated, or, like, you've learned to respond to something in such an intense and strong way, it's going to be more real to you.
Absolutely. And I appreciate whoever that is in the comments just smacking that heart button.
Yeah.
Setting up. Really quick, let's close with this. I want to know your thoughts about Elon Musk's Neuralink. I talk about this quite a bit, so I think that people generally already know that I'm not a supporter, but please, I would love to hear this from another person.
Okay. That's my thoughts on Elon Musk's Neuralink. Are you kidding me? You're taking something that neuroscientists have been doing for like 5 decades and putting your name on it and saying that you're going to understand the brain? What, you measured an action potential? Good job. People have been doing that for a really, really long time. You're not special, Elon. You just have a lot of money. You are not going to figure out what's going on in the brain unless you get some real neuroscientists to do the work. And the things he's claiming that we can do, we literally don't know enough about the brain to do. We cannot harness consciousness. Like, just like thinking about the way that the brain works is like thinking about the universe. Because if we have 90 billion brain cells, 90 billion individual ones, I can't even imagine what that number really means, like that have thousands of connections each between, so 90 billion brain cells with thousands of individual connections. I don't even know what that number is. I can't even calculate that number. And a computer these days is capable of emulating a circuit of like 10 neurons. And we've got 90 billion. And so it's like, yeah, sorry, Elon, you're not going to do it.
Yeah, Monica says, thank you for answering my sentiments exactly. Yeah, I mean, I completely agree. Exactly. I mean, implanted electrodes, being able to record, brain activity intracranially, nothing new. It's funny because like maybe 6 months ago, Neuralink published this video where they had a monkey controlling a game with its brain. And it's funny because that exact same experiment was done in humans in like 1963. And people were, and it's published, it's out there. People have known this for like over 50 years. And people were like, oh my God, Elon Musk touched the brain and now we're going to cure humanity of everything. It's like, no.
Yeah, you remember when he threw the baseball at his Tesla window, or what that truck and broke?
Yeah, the truck.
That's about where we're at. He's not got it figured out.
That was one of those transfer of pain cringe moments. Yeah, I mean, I think it's, my actual sentiment on this is that I think Elon Musk is an engineer and he will probably develop a better way to create intracranial electrodes, right? So he will help us improve our neuroscience technology.
Right, but as far as, if you're listening and you want to give us some money to do some experiments for you, I'm into that, right?
Yeah,
but yeah,
please.
He doesn't understand the brain,
right? And his comments about like the recent thing that came out where he was saying you can upload and download your dreams in like 5 years. Unbelievable misunderstanding. So
it's not. I wish it was possible. It's just not possible. That's like saying we're gonna get to the end of the universe in 10 years. Like, what does that even mean? We don't even know where the universe ends or how or if it does. Do you know what I mean? And that's kind of where we're at when thinking about the brain.
It's like the president declaring that by the end of their term there will be world peace. It's just a ridiculous thing. So, and Monica said I have MS, and people in my MS support group don't believe me that he will not cure us. Yeah, I think as far as MS, I don't see Neuralink having any involvement at all. But there is definitely a lot of great research out there that's being done. So I think there's plenty of hope, but I wouldn't attribute that to Elon Musk.
Yeah, unfortunately, I wouldn't attribute that to Elon Musk, especially he's got these grandiose ideas rather than, with MS, like he would be needing to look at like what's happening with inflammation in the central nervous system. And like, he's not even asking the right questions is part of the issue, right? But hopefully he can use all that money and sort of manpower to develop technologies that will help real neuroscientists fix things.
Right. Yeah. And I know they are hiring neuroscientists. So they got to be doing something there. But anyways, so if there are any last minute questions, please throw them in the chat. This has been a great conversation. It's been really interesting. I've been taking notes as we've been talking.
Oh, good. So you can remember the experiment ideas and tell me.
Yeah, well, that too. But like the moments where I was like, oh, this is such a cool thing that was just said. I really want to capture this and post it on like my Instagram or my TikTok or something. And I have like a long list of things that were really interesting. So I thought it was a really great conversation, and we got into a lot of cool topics.
It's probably good we finally recorded it because we have these really geeky conversations quite often.
Yeah.
So I'm glad other people hopefully enjoyed. And like I said, you want to pay tuition to the Monique and Ben Institute? Like, we can create that institute. We can chat.
Yeah, the Neuroscience of Pain and Empathy on Super Institute.
Or I don't know. We could talk about other things too. Obviously, we can go off on lots of tangents.
And can we get some thumbs ups in the chat if we think that Dr. Smith should start her own podcast, because I think that she's a phenomenal communicator. Yeah, look at all those thumbs flying.
Yay!
I really think so. I'm going to take what's happening right here as a round of applause for your excellent job.
Oh, this is ending the day in a good way for me. Thanks, everyone.
Good. Awesome. Well, I really appreciate you being here. Everyone, thank you for joining and for having such great questions. Janet, I appreciated you joining us on stage. If anybody wants to, once again, join us on stage for a moment and ask a question, feel free. We have 7 people in the queue. Again, I don't think that these people may have, I'm not sure if they joined intentionally.
Yeah.
I'm going to assume, unless any cameras turn on in the next few seconds.
Thumbs up. That's rad.
Yeah. Someone's going crazy on those thumbs up, and I love it. I really appreciate it, though. This has been awesome. And we'll have to do this again sometime, potentially.
Sounds good.
In a podcast format, although this is sort of a mini podcast itself. But
there is talk. Awesome. And I'm sure everyone's here because of your TikTok and your Instagram. But if you're not, check out Dr. Brain on TikTok. He makes really good content.
Thank you. I appreciate that. and check out Dr. Smith's awesome research, which is continuous. And she's just began her own lab. And so it's gonna be getting going soon. So very exciting stuff.
Yeah, I got mice. So there you go.
Nice.
In the lab that I started. So there we go. All right.
The experiments begin.
Ben.
All right. Thank you, Monique. It was fantastic. Thank you, everyone, for coming. And we'll see you next week. And Monique, I'll see you for another great conversation soon.
At some point soon. Okay. Bye.
All right.
Thanks, everyone.
Bye.